the opportunity to present my work. Yes. So with this seminar, I'd like to convey the main ideas of the project that I've been working along with my supervisor, Manuel Lafont, which is there in the audience. Yes, you can see him. And I would like to start by explaining the biological motivation of this problem. So first, what is horizontal gene transfer? So horizontal gene transfer is an evolutionary phenomenon, which is an important source of genetic variation, and it involves the transfer of genetic material from one organism to another. It is a really special phenomenon because it bypasses the more common parent to offspring transmission, and it is commonly seen in bacteria. But throughout the animal kingdom, there are some other nice examples. For example, it has appeared in the transmission of bacteria to animals by the coffee berry borer beetle, which transmits a gene that breaks the sugar from coffee beans. And it can also be seen in plants. And also we can see some small examples of AGT from fungi to animals. And this phenomenon is really important because it is usually related to the novel acquisition of functions in organisms. So to study this phenomenon, basically two approaches based on sequence data have been developed. First, the parametric methods, which are methods that study genomic signatures of the host organisms, for example, GC content, and when a fragment of the genome deviates from the usual, for example, in here, these outliers could actually pinpoint to possible AGT events. In contrast to parametric methods, also phylogenetic methods have been developed, which are based on the analysis of species and gene trees, which are hypotheses of the evolution of genes and of the species. We can usually study these phylogenetic methods in two ways. Either we have the explicit methods, which require the construction of genes and species trees, and they try to explain the incongruencies between them through, for example, these AGT events. And also we have the implicit methods, which bypass the need of constructing these explicitly the species and gene trees and just take a look into pairwise relationships. Nevertheless, all these methods are only based on sequences, but in one way or another. However, it has been shown that sequences actually have some important limitations when inferring evolutionary relationships, especially when highly divergent sequences are involved. It has been shown that most sequence-based methods start failing when the pairwise sequence alignment fall into something that is called the twilight zone of sequence similarity. It has been shown also that two homologous sequences can share a small percentage of sequence similarity, but they still preserve the same function. One likely cause of this is that the information that is needed to coordinate protein fold or function is encoded in very small fragments. So the main question of our work is, can we use a character-based approach to predict AGT events in a phylogeny instead of sequence-based methods? So first of all, what is a character? A character is a trait that a taxa might possess or lack. The most common example of character-based data is, for example, gene expression profiles, where a trait corresponds to whether a gene in a certain species is expressed or not in a condition of interest. We can take a look at this by building a gene expression profile, which given a set of species, for example here, and a set of genes, we can record which genes are expressed and which genes are not. As computer scientists, we abstract the results of these gene expression profiles into what we call a character state matrix, where the columns represent the characters and the rows represent the taxa, the species. And we have two binary states to indicate whether the gene was expressed or not, which I will represent throughout this presentation by this check token that we call a presence token, an on token, or you can see it also as a one, and a cross token. It can be seen as an absence or as an off state or a zero state throughout my presentation. So a well-known problem in phylogenetics that is related to these character state matrices is the perfect phylogeny problem. Well, we receive a set of characters and a set of species, which are subsets of these characters, and we want to explain the evolution of this set of taxa. We start by with a gene tree with the labeled by the taxa in the leaves, and we want to explain the acquisition of a specific character. So we suppose that we have an ancestral blank object, and our objective is to explain how the changes evolve throughout the phylogeny. So what we mean by explaining can be seen, for example, as placing our on and off tokens along a given tree in such a way, for example, that all the genes that are on remain connected, and all the ones that are not expressed also remain connected. If we can find such a way to place these tokens, and then we call it the associated perfect phylogeny. But sometimes we cannot find a way of placing our two sort of tokens along the way, so that the two sets remain connected. For example, in here, if I give you this set of taxa with these characters, if we try to connect all the off tokens, we will leave these two disconnected. And if we try to connect all the on tokens, we will leave these two disconnected. So thinking biologically, the question here could be, how could we explain the acquisition of this character by these two different clades? So in this work, we propose that this kind of inconsistencies can be explained as HT events. So to do this, we propose a mathematical structure called the Perfect Transfer Network, which belongs to the class of tree-based networks. So in here, a tree-based network is a network where we can distinguish two different types of edges. So the tree edges and the transfer edges. So tree-based networks can be seen just as trees from which we add additional edges from one branch to another. So the idea behind them is that to explain the history of a set of taxa, we could place these transfer edges to fulfill the connectivity requirements that we saw in the perfect phylogenies. So we start with placing what we know at the leaves, so the taxa and the characters, and we try to place the transfer edges in such a way that these remain connected. So to do this, we require our perfect transfer networks to follow two important rules. The first one is that once a character appears, it cannot disappear. So in other words, for a given character, if we already place a presence token along a tree edge, we cannot place an absence token, like in here, for one of its descendants. So it cannot disappear. This is an example of what is not allowed. And in here, for example, we have, like in the tree edges, we can have an off followed by an off, and an off followed by an on, for example, an on followed by an on. And the second condition is that every character should have a single origin. And by origin, we mean like the first time that this character appeared along the tree edges. So in here, for example, this could not be allowed because we could say that we have one origin here and one origin here. So this will not be an example of a PTN, of a perfect transfer network. But in here, for example, we have just one single origin, and this on token is transferred to the other on tokens as well. So if we pick an arbitrary tree-based network on a set of taxa, as I showed before, we ask ourselves, can we add labels? So can we place our tokens into this network such that every character, once it is gained, it is never lost, and it has a single origin? If we find such a labeling, such a way of placing our tokens along the network, then we have a perfect transfer network, or as I will call them, PTN. So this is the basic idea of our mathematical structure. And throughout the literature, there have been similar models to ours, but they have been used to study other evolutionary phenomena, such as recombination networks. For example, in here, recombination networks, they emulate the genetic recombination of sequences that produce new offspring. In here, we have ordered sequences, which every site represents a mutation, for example, a SNP, a single nucleotide polymorphism. And from two sequences, we create a new one that is a recombinant sequence. So we have a parent sequence that donates a small part to the recombinant sequence, and we have two parent sequences, and each of them donates a part to form a new recombinant sequence. So an important difference with this model and ours is that these recombination networks are not necessarily tree-based. And also it is really difficult to distinguish between the horizontal and the vertical descent in here. So another similar model that we found throughout the literature was in the context of language evolution. In here, they have these perfect phylogenetic networks, which emulate the evolution of languages, where each transfer actually represents a language borrowing words from another language. So in here, I give an example of a PPN, of a perfect phylogenetic network. And we see that in perfect phylogenetic networks, the transfers are bidirectional. So in our case, for AGT networks, for our perfect transfer networks, we are interested in knowing exactly who was the one that donated the gene. So direction is important for us, but in here, the transfers are bidirectional, so direction is not important. Also, a PPN contains several evolutionary trees, and a character can evolve in any of those, whereas in our model, we have a clearly defined base tree. So after defining our mathematical structure, we started thinking about its related algorithmic problems. And the first that I would like to present is the PTN recognition problem, where given a set of taxa, a set of characters, and a tree-based network, we ask ourselves, can we find a labeling that explains this set of taxa? And the answer is yes. And this can be done actually in polynomial time, which is an important difference with the model that models the evolution of languages before, because for these PPNs, this problem is actually in p-hard. So an important observation to solve this question is that if a vertex V has at least one descendant with an absence token, with an off token, then V should also be an off token in every possible labeling that explains S. To see that, suppose that it isn't. So suppose that you have a descendant that is that has an off token, but you are an on token. Then it would mean that a character was gained and then it was lost. So it violates our definition of PTNs, one of the rules that we established at the beginning. So we see that there are many the nodes that fulfill these conditions. So the nodes that have at least one descendant, which is an off token, or we do not have a character, we call these nodes force zeros. So for example, here I have another example where we can see the effect of force zeros. So in here we have two networks with two different labelings, so two different PTNs. And in here we see that the force zeros are the ones that will always be a zero, no matter which labeling. So in here we see that we fulfill the condition of having a single origin, we can take the origin in here, and we still have all the on tokens are connected, and all the off tokens are also connected. And if we try this another labeling, for example, if we place this token as an off token, we can still pinpoint an origin for all the on tokens, and the 4 zeros will remain the same, will remain as off tokens. So using 4 zeros, if we proposed a lemma, we proved a lemma where if the following is true for every character in our problem, then we can say that an arbitrary network is a PTN. So this is the high-level version of the lemma. So we say that G is a perfect transfer network if and only if, when we remove all the 4 zeros, we can find a node that reaches every leaf marked as an on token. So I'd like to present a small draft of this proof. So for the forward direction, we start with G being a PTN. So if G is a PTN, we already know how should we place the tokens along this network. So in here we see, as I've shown before, that the 4 zeros actually belong to the set of off-labeled nodes. So the nodes marked with an X. And when we remove all this set of 4 zeros, then we will have a component that still has off tokens and on tokens. But by the single origin requirement of our definition of perfect transfer networks, we can find a node V that actually reaches every other on leaf. And in the other direction, we need to build a labeling that fulfills our definition of perfect transfer networks. So this direction actually implies the algorithm that will serve us to identify whether something is a perfect transfer network or not. In here, to build a labeling, first we start by identifying the 4 zeros, and these ones are marked as off with our off token. After removing the 4, an important note is that after we remove the 4 zeros, if we get a disconnected network, then this means that we cannot reach every on token. So this could not be a perfect transfer network. But if we get a connected network after removing all the 4 zeros, then we should be able to find a node, V, that reaches every on leaf, for example, in here. So the strategy is, okay, pick this node that reaches every on leaf and mark every reachable node as an on. And if we do this for every character, then we can verify in polynomial time whether something is, whether an arbitrary network is a perfect transfer network or not. So another interesting algorithmic problem is explaining the history of a set of taxa using the transfers. So you're giving suppose that you're giving a base tree and a set of taxa with a set of characters, and the goal is to add transfer edges to explain the evolutionary history, which is the question that we had at the beginning. So an important part of adding transfer, if you have worked with AGT before, then you know that things can get complicated when we take time into consideration. So we know that AGTs cannot actually happen at arbitrary points in time because we need the two species to coexist at the same time. So a mathematical way of ensuring that this transfer remain time consistent is that we know the transfer actually creates cycles. So if we add transfers in such a way that we do not create cycles in our networks, then our networks will remain time consistent. So an important observation before solving this problem is that every given network actually has an implicit labeling for the given tree. So we can see this as a sort of Fitch labeling. So when we assign every inner node a label according to the label of its children. For example, if you have a children that is an on token or an off token, we will assign an off token. And we will only assign an on token when both of your children are on nodes. So in this way, we end up with a tree that already has some implied labeling in there. This labeling actually tells us which characters could have appeared first. So whenever we have a node whose parent node has an off token, but all of its descendants are marked as on, then we call this a first appearance node. For example, in here we have 3 first appearance nodes. So these 2 leaves that fulfill a definition also, and this one. So we have 3 first appearance nodes in here. A simple way of adding transfers to solve our problem would then be to join every first appearance node in a time consistent way. So what do I mean by in a time consistent way? So what we can do to ensure time consistency is first, suppose that we're giving also a time map, so we can order all the first appearance nodes chronologically. So we assume that every node is assigned a distinct time in such a way that in this ordering, we can know who is bigger than who. For example, in here, we could say that X is bigger than Y and Y is bigger than Z. So the idea would be to join every node with its successor or with a suitable descendant of its successor. So for example, in here, if we join X and Y, what we do is that we subdivide this edge, and we join the 2 first appearance nodes, and we mark this path with on tokens as well. If we join every first appearance node this way until they are all connected, then we are done, and our problem would be solved. So notice that the algorithm that I explained in high level is somehow suboptimal. And I will show you why using 2 characters. So suppose that that we have 2 characters, a pink character and a purple character. And we have our tree with the pre-labeling that I explained before. So suppose we started in here, taking this first appearance node to explain the evolution of the pink character. So if we look for the other first appearance node, which would be here, then we will create some sort of path between them. And then when we try to explain the evolution of the purple character, then we will end up adding 2 additional transfers in here. But notice that in this way, the optimal solution, in this way, we are using 3 transfers, whereas an optimal solution uses 2 transfers for these characters. This is just only due to the randomness of our algorithm. Actually, this leads us to an important open problem. So some open problems that are left from this from our study is, can we find a perfect transfer network that actually minimizes the number of transfers? And also, given a species tree, can we add a minimum number of transfers so that it explains the set of taxa? We still haven't been able to prove this, the complexity of these questions, but our conjecture is that the 2 is probably NP-hard. So as a future work for our model, we know that now we're using perfect characters, and we've seen that these perfect characters actually impose strong restrictions to our model. So we would like to change the conditions on how is it possible to change from a 1 to a 0, for example, as in the dollar parsimony. So in this way, just adding some other models of when can we add, for example, losses to our model. And also in the future, to instead of having binary states, also include different expression levels for each character. And if you are interested in this work, I invite you to take a look at our paper, which can be found here. And that's it. I thank you all for your attention.